So continuing our class on crop improvement, we'll see other important breeding techniques. So herein, today we shall look into the types of the mutations. Okay, how mutations are involved in breeding a good variety of crop. So by definition, we know mutation is a sudden heritable change. And we are making use of this mutation to select a desirable trait in our plant. So you know that Ugodi virus observed mutation in evening primrose plant, and later on Muller introduced mutation in Drosophila by using the X-rays. So there are two types of mutation. One is the spontaneous mutation and other is the induced mutation. So spontaneous mutation are natural mutation. That is, they are caused by themselves in the nature, and it is very low in frequency. Whereas induced mutation, low in frequency, means like what we mean to tell is like when the mutation is happening, when the changes are happening, we'll not be knowing it very immediately. Then the induced mutation is then artificially induced by man. Okay, like for instance, the X-rays. How X-rays were used by Muller to induce mutation in Drosophila. That is one of a classical example of induced mutation. So now the mutation can be macromutation or micromutation. Macromutation means they are visible. The morphologically, the changes are visible. Phenotypic changes are visible to us. Whereas in micromutation, the morphological changes in the phenotype is invisible. Okay? So, like how we are going to induce mutation? The mutation most probably what we use for the crop improvement is the physical and the chemical mutations, and the agent which induces these mutations are called as the mutagen. And this one you can say that induced mutation is quite fast when compared to the spontaneous mutation. Okay, in short period of time we can see the output. So in physical mutation, we commonly use ionizing and non-ionizing radiations as we have mentioned here. And even in chemical mutagens, like commonly what we use is the colchicine, acridine dyes, methane sulfonate, all these chemicals are commonly used. So, inducing desirable mutation and exploiting them in the crop improvement is known as the mutational breeding. So, we are using the mutation technique to bring about some changes in a plant crop, desirable changes, not unwanted ones. Okay, when we're using this mutation technique to get desirable variety in a crop plant, then it is known as a, this phenomena is known as a mutation breeding. Now whenever you are going to do a mutation, first what should strike to our mind is like we have to select the best variety or the best variety of seed and then proceed with the mutations. Like this physical mutations, whatever we are telling, like this X-ray or UV radiations, these are particularly used for the vegetative part of the plant. Whenever we want to bring a change in the vegetative part of the plant, preferably we use physical mutation, whereas the chemical mutations or mutagen are preferred for seed treatment. So these are the two different um, mutagens. And the physical, just remember, it is used for the vegetative part, and the chemical mutagens are preferred for the seed treatment. So almost to get a desirable variety, it almost takes 9 years. Okay, because here we do not want any unwanted or undesirable changes to propagate in a plant. So it takes about 9 years to obtain the mutant variety. And that is, the scientists will do their experiment in their substations, grow the plant, okay, select the best variety of it, and screen for the best phenotype finally, and then commercialize these. So in the year 1970, you can see this International Atomic Energy Agencies. Okay, these agencies, what they did was they subjected many plant varieties to mutagenic agents to induce mutation, and then they selected the desired new traits which appeared, which was favorable trait to propagate that plant. So this induced mutation assisted breeding, okay, has resulted in the introduction of new varieties of many crops, particularly rice, wheat, barley, which are commercially available in the market. The drawback when we are talking about mutation is we have to see that the mutagenic agent is not passed into the food item. So it should get clearance for the Food Administration Organization, so that it does not cause later on any deleterious effect on the environment or on the people who are, or whoever is consuming that particular type of food. So this we should take care of, so that whenever we are inducing the mutation, we have to keep in the mind like what dosage is required, and a protocol is framed and they proceed according to it. Coming to the next type of breeding is known as the polyploidy breeding. So again, you have to recall your genetic classes here, which we have dealt what are polyploidy. So polyploidy, you know that there are two or more sets of chromosome is known as polyploidy. This can occur naturally or one can induce also. So utilizing this technique of polyploidy for improvement of crop is called as polyploidy breeding. And polyploids are plants are available naturally also. Like almost all your grasses, which you see, is all polyploids. Okay, and also this endosperm of the plant, it's a triploid. The wheat variety is a hexaploid. Okay, hexaploid means 6N. Hexaploid. And strawberry, okay, it is an octoploidy. These are some of the natural examples, and many more are there. So the types of polyploidy, what we see is the triploids, that is 3 sets of chromosomes, tetraploidy, 4 sets of chromosomes, and there are also autotetraploids and allotetraploids. Um, just to recall what we studied in our genetics, what are this auto and allotetraploids? These are these how these are formed means like whenever there is non-disjunction. Non-disjunction means that is the failure of the chromosomes to separate. This results in the subsequent reduction in the chromosome. Okay, so that what happens, all the chromosomes just go to one cell, there is no separating. So this can result in the polyploidy. And some of the important polyploidies which are helpful in crop improvement are this autotetraploides and allotetraploides and the others whatever we spoke about. So if you recall our genetic class, we told that polyploidy may not be useful for animals, but in plants they have their beneficial effect because they can give a very good output product. So this autopolyploidy, here you can see this, here this is the example of autopolyploidy, that is the doubling of the same chromosome set. That is, autoploidy are derived from the within a single species. So from a single species itself, we can double the chromosomes and we can get the output. For example, you can see in this example, okay, what is this? A diploid cell. So your mitosis happens, and suppose say there is failure of cell division here. Okay, failure of the cell division after chromosome duplication. What happens? It results in tetrapolyploidy. That is, it has just doubled itself because there was some error in the mitosis. So a diploid has become a tetraploid. And then this, when it undergoes meiosis, then again you get a diploid gamete. And after fertilization, you end up with a tetraploid. So this is called as autopolyploidy. That is, it occurs when an individual has more than two chromosome sets derived from one species. Okay, just remember this. It is only one species here. What I mean to tell is, suppose say this is A, okay, A and A. So this is what it is, a triploid. And these are all same alphabets what I have written. Okay, that is same species. Or if I write it as A. So what is this? It is a tetraploid. It is an example of autotetraploid because of the doubling has occurred in the same species. Hope now it is clear. So here what has happened? A diploid cell in mitosis can become tetraploid because there is a failure in mitosis. And meiosis, what it has yielded? It has yielded a diploid gamete. A new species arises with a fertile tetraploid offspring. That is your autopolyploidy. In allopolyploidy, what happens? It is also called as amphidiploid. Okay, this one. So, allopolyploidy, it is a product of interspecies hybridization. That is, there arises between two different species. Here, within the same species, here two different species. That means, for example, let me write it here and show you. A, okay, then B. What it tells, this is what it is, a tetraploidy. But what type? It is two different species. This is a two different species. So these are called as allopolyploidy, or it is called as amphidiploid because it is formed from two different species, hybridization of two different species. So this is another example. See here, species A, species B. So it has 4 chromosomes here, it has 6. And we can see here there is an error. So what it has unreduced gamete. So whatever here the chromosome number is, same number is here. Whereas here it has reduced. So it is a normal gamete. So only 3. Okay. Then when hybridization happens between these two, we get an hybrid which is having the 7 chromosomes. Okay, if you count, it will be 7. Okay, so the blue color you can see the normal variety, and here it is unreduced, it is not separated. Okay, non-disjunction, and here you can see that it is unreduced gametes are being formed, and whatever the other three normal gamete was there, that one we have put it here, and then after hybridization, what we get? 7 plus 3, we got 10. So this is an allopolyploidy. So what has happened? Your chromosome numbers are doubled. That is 2 or more sets of chromosome. Here also, here also. But here it has happened in one species. Here it has happened between the two different species. That is why it is allo and this is auto. So this phenomena we can use it for the crop improvement. Next, micropropagation, another important technique which we shall also study in the further classes, future classes, more detail about this. But let us give a touch upon what is micropropagation is all about. Now, this micropropagation, in other terms, it is a plant tissue culture. Okay, here it is being practiced for rapidly multiplying stock plants in vitro under aseptic condition to produce many progeny. That is one of the output of our micropropagation. That is, we are developing or growing the plants in vitro, that is in laboratory. Here you see in the glass vase they are grown. When you provide the media and the proper environmental condition, they start growing, okay, under the aseptic condition, so that we can get many progenies. So this, as said, micropropagation is particularly useful for maintaining speeding up of plant breeding, rapidly developing, multiplying the plants, so that we get more number of the plant material. The last one is genetic engineering. So as you know, the meaning of what is genetic engineering is just to introduce a foreign gene into the cultivar or the crop of interest. This foreign gene, whatever we are telling, it should be a gene of interest, that is the good genes. So it is introduced into the plant, and so that whatever the good genes are there, it gets integrated into your plant of choice, and we get the best variety. So genetic engineering in crop improvement, we can use three techniques. One is the close transfer, that is taking a gene from one plant species and inserting it into the another plant species of the same kingdom. Another is the distant transfer, taking a gene from one species and inserting into another species from different kingdom. So it may be a bacteria. From the bacteria, you take the gene and put it into a plant. So what happens? They are both two different kingdoms. One is plant and one is the microorganism. Or the other one is the tweaking. Tweaking means you make a small adjustment in the gene, or you fine tune the gene, okay, so that it produces the best variety of the crop. So what happens if a gene is there and you know that gene is responsible for producing some of the best trait, okay, but you still want the more best out of it. So what you can do is slightly you can modify this gene. You make a slight adjustment, just do a fine adjustment of this gene, okay, so that genes will be treated to change the level at which the protein is made, so that we can get again the best variety of our crop. So all this genetic engineering, whenever we do, Okay, it gives us the genetically modified food product or the organism. One example I just wanted to tell you all is like the researchers from JNU, India, and also the National Institute of Plant Genome Research. They had a campaign that they wanted to see to that the children do not suffer from any deficiency from the protein. So what was their idea is like we all, most of us like potato, and we know that potato is rich in starch and it has small amount of protein also. So these group of people from JNU and the other institute, they've come forward, and using this genetic engineering technique, what they have done is they have transferred the gene from the amaranth plant. Okay, see here, this is the amaranth plant. They've taken the gene from this plant, the grain of this plant, and they have transferred into potato. So this plant is rich in protein. Now, when they have transferred into this has potato has become genetically modified. Now, this potato, now which they have named it as potato, 30 to 50% more protein is present in this, the new variety. Okay, and it consists of very good amino acids also, like lysine and methionine. So this is one of the good example, like how they have modified by using genetic engineering to get a very high efficiency of protein and the essential amino acid, almost like 30 to 50% increase in the potato, you can see. So these are the some of the techniques, okay, like plant introduction, mass selection, hybridization, heterosis, polyploidies, mutation, micropropagation, genetic engineering techniques. All these are utilized for the betterment of the crop improvement. So what we told in the starting of our class, okay, our aim is to see that we get better crop, better food variety with high nutrition, so that it can solve the problem of hunger. And one more, like we have this gene banks have been created. Okay, that is, they are like repository. Repository means where you can store. Okay, like biorepository, that uh, there is a presence of genetic material in those plant. So now what happens is like whenever like a scientist needs some plant which is having a particular trait, he can just go to this bank, whatever the gene banks they have created, take that plant, and they can cross it with another plant which is having a second desirable trait. Okay, so this already a plant which is having a good trait or a beneficial trait or a desirable trait which is there in the bank. Okay, he takes that and he crosses with another plant which is also having another desirable trait. So again, what we are doing, we are doing a breeding technique which is better than our traditional or conventional methods. So this concludes our topic in crop improvement.